Hello everyone, thank you for the kind introduction. I will briefly introduce you to the antimicrobial resistance crisis, and I will inform you what we do as our contribution to the global call of new antimicrobials. To introduce to you the antimicrobial resistance crisis, I show you this snapshot of a tweet. This tweet I've sent out 2 years ago under the hashtag of the 10 years challenge, and this tweet really went viral. What you see on the left-hand side is a plate with bacteria, and all these white discs on the plate contain a different antibiotic. And you see this transparent halo surrounding every disc. This means that the antibiotic is still active. It kills the bacteria cannot grow. That's why you see this halo. If we take a strain of the same species, but we go 10 years later, we see that many of these antibiotics do not work anymore or have a reduced activity. And that's what's going on. An increasing number of strains get extensively drug resistant or even pan-drug resistant. The latter means there is no single antibiotic available on the market that can be used by the doctors to treat an infection with this strain. And this means that only your immune system can cure you from this infection. So this problem is on the rise and challenging more and more our global healthcare systems. There's not only alone the increasing emergence and spread of resistant bacteria. At the same time, in the last 2 decades, there was a declining interest of the pharmaceutical companies in antibiotic discovery. It was not a sustainable business plan. It was much more interesting to invest in drugs and medication that you have to take a long life, the complete life, like for example, for cholesterol, for example, than to invest in antibiotics that only have to be taken a few days. So we end up in a kind of perfect storm where at the one side you have the spread of the resistant bacteria, and at the other side there are too little new antibiotics approved for introduction in the clinic. And this will eventually end in a kind of pandemic of antibiotic-resistant bacteria. If we do not take action, a very frequently cited report predicts that we will have 10 million deaths per year by 2050. And to put that in perspective, currently we have about 3 million deaths due to corona. So this number is even higher. Also, if you calculate this in terms of the effects on the economy, there will be an accumulated loss of $100 trillion by 2050. There is a second problem related to antibiotics that I would like to emphasize here, and that's the use of broad-spectrum antibiotics. A broad-spectrum antibiotic does not only kill the bacterium that makes you sick, the pathogen, but it also kills other bacteria that are benign, that are part of your flora in the gut, on the skin, and so on. And there is this famous book published by Martin Blaser, who wrote about the missing microbes that are lost due to the action of broad-spectrum antibiotics. And there is increasing amount of evidence that these missing microbes contributes to all kinds of modern diseases that become evident later in lifetime. So globally, there is a need for new classes of antibacterials to counteract this emerging spread of antibiotic-resistant bacteria, but at the same time, there is a global increasing interest in micro-friendly antibiotics that do not kill the friendly bacteria. So we need narrow-spectrum antibiotics. Our contribution focuses on the use of viruses, and this has been covered recently by the first edition of our UGent magazine with the tempting title, Can a Virus Rescue Mankind? And this is, of course, a very strange title maybe, because in these times of corona, we think viruses can only be bad, but here we are speaking about bacteriophages. Bacteriophages are the viruses of bacteria. They are the enemy of bacteria, and the enemy of our enemy is our friend. Okay. Here you see a cartoon of bacteriophages. They have a head, a capsid, a tail, and tail fibers. And these tail fibers are kind of antenna. They look for bacteria, and they look, is this bacterium the right bacterium? This interaction must be very specific. And bacteriophages are very specific only for a limited number of bacteria. They're specific at the strain level. Only when the receptor is on the bacterial surface and interacts with this fiber, there can be an infection. And after an infection, the bacteriophage replicates inside the cell. You get about 10 to 100 copies of the phage particle, and then at the end the cells are lysed and broken and killed. And this has led to the concept of phage therapy. What you see here are bacteria that make us sick. And phages have been added to this bacteria. The phages are labeled green, and this is what happens if you start the infection. So you see that the bacteria multiply, but at the same time the first bacterial cell starts to lyse, and you see that the phages come free, and it's in fact a kind of firework. And you see how the phages are replicated in the cells, and how they spread and disseminate. And this is the concept of phage therapy. It's an old concept, it's more than 1 century old. And there are a number of very interesting case studies where you see that phages could cure patients. Here you see maybe the most famous case study with Tom Patterson. Tom Patterson was a professor in California, and also his wife was a professor. They went on a journey, on a travel in Egypt, and he was hit by a palm drug resistant bug. And even the most modern healthcare systems could not help him. He was dying. There was no antibiotic that could cure this Tom Patterson. And then his wife looked for phages. She collaborated with phage researchers, and they composed a phage cocktail, and Tom Patterson could be cured. It was almost a miracle, but it was a case study. You can read about this in a novel, The Perfect Predator, but at the same time there will be very soon an episode on Netflix on that, and also a Hollywood movie is currently recorded. So it's a very famous case study. But at the same time, they're very high profile clinical phase studies, and phase 2, where they test the efficacy on the broad population of patients. And there you see that even with the best efforts at the moment, that we fail. It's very hard to develop phage therapy as a general solution for all kinds of patients. No, instead, it remains an ad hoc strategy, and one always has to select the right phage for the right patient, because phages are very specific. From one hand, it's very good because they're microbiome friendly. They will not kill other bacteria from your gut. But from the other side, it's very difficult, because for every patient, you have to isolate the bacterium. And you have to look for the right phage in the phage collection that you have. So this takes time. So you can only use it for chronically infected patients. Another solution is the use of cocktails, that one composes one makes a cocktail of different phages together, and all these phages cover all the different strains that can occur. But this takes time. It's difficult in terms of production, but also in formulation and stability of the cocktail. So these are true hurdles of phage therapy to roll it out as a broad way of treating patients. In addition, every new phage that you isolate and that you want to use for a patient has to be studied in detail, because some phages cannot be used because they have toxins, or they are lysogenic, so they're not killing the bacterium immediately, but they integrate in the genome. So what's our contribution to these hurdles of phage therapy is that we have developed the phage engineering platform. Instead of looking for a new phage anytime, we start from a phage that's safe, and we use that as a phage chassis. And instead of looking for a whole new phage, we only exchange the tail fiber of this phage. So we cross the chassis phage with another phage that is specific and has the right tail fiber, but we transplant this tail fiber on the chassis, you end up with this altered phage. So we rely on the well-known chassis, but we just change the specificity. By exchanging the tail fibers, this is here visualized by the colors, you can make them specific for a broad set of bacteria. So we can make a phage cocktail where all the phages have the same chassis, but only the tail fiber is exchanged. And then you don't have to study every new phage because the tail fiber is always a safe protein that we transplant in the new chassis. Here you see an experimental result. So we have Klebsiella phages. Klebsiella typically have a receptor, have the capsule surrounding the cell as a receptor. It's a sugar layer surrounding the cell. And you have phages that infect bacteria with the K3 capsule in light blue, the K11 capsule in light gray, or the K21 capsule in dark blue. So what did we do? We started from K11. That's our safe scaffold phage. That's the chassis that we used. And then we transplanted the tail fiber of KP32 to the chassis. And what you see is that instead of infecting K11, the specificity switches to K3. And we could do the same with this tail fiber, K21. We transplant it to the K11 scaffold, and you see that the specificity switches to K21. So in this way, we can make a phage cocktail with conserved chassis, a conserved scaffold, and we only transfer the tail fiber. This happens in nature. This is in fact a natural process that we mimic now on the lab scale. And we convert it in fact to a pluggable system in a conserved phage chassis. A second aspect that I want to tell about are the weapons of bacteriophages. When a phage replicates in a cell, you get a lot of new phage particles. These are the white particles on this electron microscopy image. And at the end, the phage will produce a protein. It's called the phage lysine. Here you see a crystal structure of it. And this degrades the cell wall from the inside. And when the cell wall is degraded, the cell will explode because there is a high internal pressure inside the cell, an overpressure, and the phage particles will be disseminated and they can look for new potential host cells. So these phage lysines are very powerful enzymes, and we found that when we purify them and we apply protein engineering and then apply them from the outside to pathogenic bacteria that makes us sick, that they work as an antibiotic. And this is what you see in this movie. This is Pseudomonas aeruginosa. You add our engineered phage lysines to them and all the cells are killed. You see that they lyse and they explode like in the movie of the phage therapy. The nice thing of these engineered phage lysines is that they're also very specific, but not as specific as phages. They are maybe too specific, but here the lysines are specific at the species level. Here you see spherical cells. The spherical cells are the good bacteria, part of our skin flora, while the rods are the bad bacteria, the pathogenic bacteria. Here we apply a phage lysine that's very specific for the rods, and you see that they're killed immediately, and only they are killed, while the spherical cells remain intact. So we kill the pathogenic bacteria, but the good bacteria remain unaffected. So it's a micro-friendly antibiotic. So lysines are a new class of antibiotics. They're currently evaluated in the phase 3, so that's the last step before market approval. But they have a very interesting property. An interesting property is the modularity principle. It means that there are many possible lysines that can be made and tested because they're composed of building blocks. You have building blocks, I will not go in detail, but you have EADs, CBDs, peptides, and linkers. And you can make an endless variation of combinations. These are modular proteins composed of different building blocks, and every composition has a different antibacterial property. You can change the specificity, but also the activity, the production yield, and so on. And the challenge is to make a lot of variants and to find the needle in the haystack, to find this lysine, this modular lysine that kills your pathogen at best. And that's what we do. We created a new technique. It's called the versatile engineering platform. And this is in fact a modular approach that mimics Lego. We all know Lego. You first have to buy your box with building blocks, and then you can stick them together. You can make your any construct. And that we do at the protein level, at the DNA level, encoding for these phage lysins. First, we make a repository with all the building blocks. We call them tiles. And once we have this repository with all the tiles that are these EADs, these CBDs, these peptides and linkers. Once we have this repository, we can stitch them together in many different ways. And we can make all the variants needed, all these variants that you see here, but the numbers we can create are tens of thousands or even millions. And then the next step is that we have designed screening assays to find the right lysine that kills your bacterium. So we take a bacterium from a certain patient and then we screen which one is the best. And you see the majority of them is not working well. So what you see here on this graph is how well the cells grow. So the lower the bar, the better the bacterium is killed and inhibited. And you see from the screen of 96 variants, there's one, the C8, which completely inhibits the growth of the cell. So this is the good one. This is the needle in the haystack we were looking for. You have 3 other ones that also partially kill the bacteria, but they have only an intermediate effect. So this is a good example how you can find the right lysine from a broad collection of modular phage lysins. So then I come to my conclusion. The 2 take-home messages are that bacteriophages and their lysins represent a versatile class of antimicrobials and that they can potentially target any potential pathogen. So it's a very broad class. And we blend these natural solutions with approaches from the field of synthetic biology and protein engineering to really make customized micro-friendly antibacterials for any possible pathogens. So the enemy of our enemy is our friend. I thank you very much for your attention.